and I'll be going over cell biology, which is the topic of my talk. I don't really have a preference if you turn on your camera or not, but I will ask you to keep yourself muted. And if you have questions, you can, of course, unmute yourself, or you can just type it in the chat. Okay. So my name is Ethan Kern. I'm currently a freshman from Dallas, Texas in Cistercian Preparatory School. And I obviously enjoy biology, and my hobbies are robotics and playing the cello. All right. So first of all, how scientists study cells in the first place. So the development of instruments that enhanced human science allowed the discovery and early study of cells. Microscopes themselves were invented in 1590, and they were much improved in the 1600s. So this guy called Robert Hooke discovered the cell in 1665 when he looked through a microscope at cork. So like the cork from like a wine bottle, which are made of dead tree cells. He didn't see living cells until he visited Antony von Leeuwenhoek, which I'm pretty sure I butchered the pronunciation of, in 1674. And he called them very little animalcules. So the first microscopes that were invented were called light microscopes, abbreviated as LM, where visible light is passed through the specimen, so like the thing you're looking at, and then through some glass lenses. These lenses bend the light in a way that magnifies the image, so like hundreds of times. These are probably the microscopes you have at your school in like a middle school or high school. And then later in the 1950s, the electron microscope, or EM, was introduced to biology, of which there are two types, scanning and transmission. Scanning shows the surface of the cell, while transmission shows a cross-section. The EM could magnify hundreds or thousands of times more than LM. However, a major weakness of the electron microscope was that cells were required to be killed before use. I think some EM have a requirement where you have to cover the cell or specimen in gold before you can actually use it. And here I have a picture, oh wait, back. I have a picture of an electron microscope, which is obviously a lot bigger and more complicated than your average school microscope. And yeah, that's how scientists look at cells. All right, so what is a cell in the first place? So a cell is the smallest unit of life that can function on its own. All living things are comprised of cells. So all cells share four characteristics, and they are a plasma membrane made of phospholipids. So if you can see here on the left here, we have a cell. So like the circular thing, like the outer shell, is called plasma membrane. And then the cytoplasm, the liquidy stuff surrounding the organelles inside the cell. So the organelles are each individual little section inside the cell, and then the cytoplasm is basically the rest of it. Then DNA, and then ribosomes, which are like tiny, tiny little structures that make proteins, basically. All right, so the cell membrane, and why is it so important? So the cell membrane surrounding each cell forms a barrier between the cell and the outside world. It mainly regulates the passage of substances in and out of the cell to help maintain homeostasis, which is the tendency not to stray from the ideal conditions for the cell to work. So like some things in the cell have a specific pH value, a specific temperature, or a concentration of like sodium, for example. So the cell membrane helps regulate that so that It'll never be too high or too low. The cell membrane is made up of phospholipids, which I have a picture of here. And you can see that the circle on top is called a hydrophilic head, which is attracted to water. And then these two little stick things at the end are called the tails, which are hydrophobic and repel from water. All right, so when phospholipids are put into an aqueous environment, they automatically form a sphere. Does anyone have any guess to why? You can unmute yourself or type in the chat.
Because of gravity?
Not really because of gravity. Not surface area.
Hydrophobic.
It does have to do with hydrophobic and hydrophilic.
Wait, which one though are you talking about forming into?
This picture here, if you can see the picture.
Because the hydrophilic is attracted to water, so it's on the outside of the sphere, and the hydrophobic repels from water, so it's in the inside.
Here's the problem with that. It's a really close solution, but the inside of the cell also has water.
It's the cell membrane is made up of 2 layers of phospholipids.
Yeah, that's the correct solution. And all right, so the phospholipids will form a double layer, which you can see here with this picture. So the outside of the outside of the cell membrane is water. So like maybe blood, for example. And then the inside, the cytoplasm is also mainly water. So the hydrophilic heads face the outside and the inside, and the hydrophobic tails face each other. And because these two, the hydrophobic tails do not want to face the water, they automatically face each other. And scientists have a fancy name for this, as they have for everything, and it's called the phospholipid bilayer. Does anyone have any questions about this? All right, moving on. So the cell membrane are embedded, membrane proteins are embedded throughout the bilayer, and different membrane proteins carry out different functions such as transport, recognition, and adhesion. One example of this recognition thing is cell type. So blood type. We have AB, B, A, and O. And what those really mean is what type of membrane proteins you have outside of your blood cells. So some people have A, so you have the specific protein, some people have B. O means you have neither, A, B means you have both. And some proteins and phospholipids in the cell membrane have chains of sugars attached to them, or carbohydrates. These play a role in communications between cells. And cholesterol is an example of a membrane steroid. Now, remember, for steroids, I'm sure that some of you, when you hear steroids, you think of Arnold Schwarzenegger steroids, where it's like anabolic steroids, we get tons of muscle and everything. Those are called anabolic steroids. Steroids basically mean like any molecule that has like this specific structure of 4 carbon rings. And that's pretty much just it. So the membrane steroids keep the membrane at the right level of fluidity, so it's not too soft or not too stiff. So the cell membrane is neither solid nor liquid, and it's more like jello. So how that works is, if you look at this diagram here, so in the membrane, there's like cholesterol in it. So it's like some, how do I put this? It's like a joint, just many little joints throughout the cell membrane, so you can like adjust the fluidity. So if you have like a lot of cholesterol, it's like very fluid, and if you don't have enough, it's like way too stiff. So in the membrane, the phospholipids do not stay in one specific place. They're always constantly moving and switching places. And so this is called the combination of phospholipids and proteins. It's called the fluid mosaic. And also plant cells have an additional cell wall that provides additional structure. Are there any questions about this part or any explanation of this diagram that you need? You can type in the chat too.
What type of things would the different membrane proteins move into the cell?
They will always face, like these sugar molecules, they'll always face outwards of the cell, never inside, because if they were facing inside, they wouldn't have much purpose because nothing would attach to them. And
No, what is for the membrane proteins that do the transportation in and out of the cell? What type of things do they accept into the cell, and what type of things do they not allow into the cell?
okay, so the phospholipids, they typically let in lipids, so basically the same type of cells, so like fats, that kind of thing, so lipids. And also they let in small non-polar molecules. If you know what that means, it's basically like carbon molecules like carbon dioxide, where the electrons in the atoms of the molecule are evenly dispersed. So like an example of a molecule that would not pass through is water, because it is the electrons are not evenly dispersed. So small non-polar molecules and lipids would pass through.
Okay, thank you.
That's not to say that water will never pass through. It's that it needs the help of some protein channels that help it go through the membrane. And sometimes you even need like energy to really force it into the cell. So like sodium and potassium, you do need energy. So cell theory. So basically, like centuries after the cells, they started to find some similarities between cells. So what they discovered early on is that all organisms are made of one or more cells. Cells are a fundamental unit of life, and all cells come from pre-existing cells. And some things we discovered later are that all cells have same basic chemical composition. So basically, all cells basically use oxygen, carbon, nitrogen, sulfur, phosphorus. And all cells use energy to do work. And all cells contain DNA that is duplicated and passed on as the cell divides. You can unmute if you have a question.
If all cells come from pre-existing cells, how did the first cell form?
That's a very good question. So there's an experiment, if I recall, that replicated conditions from billions of years ago, and out of that came cells that did not exist before. And I'm not too sure about the specifics of that experiment, but you can go look it up after if you're interested in that.
Okay, thanks.
All right, no problem. All right, is that it? Okay. So why are cells so ridiculously small? So like humans have trillions and trillions of cells. So basically, the main reason is that smaller cells have more surface area relative to their volume. What I mean here is suppose there's a question in the chat. Oh, no. Suppose I have like a cube with a 1 unit long edge. It will have a volume of 1 and a surface area of 6. Okay, let's compare that to a cube with a 2 unit long edge. It will have a volume of 8 and a surface area of 24. And the ratios between these are very different. So it's 1 to 6 for the first one and 8 to 24 for the second one, which is basically 1 to 3. So why is this important? So high surface area allows the cell to quickly exchange materials with its surroundings. So basically what we mentioned earlier with the cell membrane and how different molecules pass through. And for example, oxygen from the air has to enter your lung cells pretty quickly or you'll die. And a cell with more surface area, but with a smaller volume, will be able to intake more oxygen than one with small surface area and big volume. So it's really all about efficiency. And a very extreme example of this is like the nerve cell of a giraffe, which has to extend from its body all the way to the head, and it has to span the entirety of its neck, which is like super long, right? And so it is many, many times thinner than your typical cell, and it's 15 feet long. Like one cell is 15 feet long. So, all right, do you have any questions about this part?
So for giraffes, how do they transport the oxygen then? If the cells don't get oxygen very quickly, then they'll die. The giraffe's neck is extremely long. How do they absorb oxygen then? Because it has to go through the lungs, right?
Yeah, but this is a nerve cell.
No, like all giraffe cells, not just a nerve cell.
So I think the nerve cell is just the exception to that. And how they breathe is just there's just lots of cells that transfer it to the lungs and then all the way back. So they have like a throat, so they breathe in oxygen. The throat is just really, really long, connects to the lungs, and they just breathe it out.
Okay.
I'm not sure if that answered your question. Is that all right?
Yep.
All right. So we'll get to that later, those messages in the chat. All right, so prokaryotes and eukaryotes. So cells are classified into 2 very general groups. So they're called prokaryotic and eukaryotic. Prokaryotic cells are what the first cells are. So they're the most ancient forms of life, literally billions and billions of years old. And they're super small, simple in structure, and they don't have a nucleus. So one example would be bacteria, which I have a picture of here, which is basically some ribosomes which just make proteins, DNA floating in cytoplasm, and cell membrane. And that's it. Maybe they'll have like a flagella, which is the little tail you see at the end, and the cilia, which are like the hairs. And then eukaryotes, which are what we are made of, is very evolved billions of years after prokaryotes. So they're relatively new. And they're thousands of times larger, more complex, and many internal parts, including a nucleus and other organelles, such as the mitochondria, the powerhouse of the cell. And I have an example of a eukaryote here. And then, unlike prokaryotic cells, so the main difference between them, besides their size, is that they divide the labor between organelles, which do everything from protein production to digestion. So an organelle is basically like a little part of the cell that has like a different purpose. So each organelle has like a different purpose. A mitochondria is one of them. They do energy stuff. And then the nucleus, etc. Is there any questions about that? In quotes. All right. So this is basically a simplification on how proteins are made. So the nucleus contains DNA, which specifies the recipe for the proteins, and it also contains the nucleolus, which produces the ribosomes. And RNA is made in the nucleus, and each RNA, so basically we're using mRNA here, is specific to each DNA. So like every sequence of DNA has like a unique RNA by base pairing, you know, those A, T's, C's, and G's. So A matches with T, C matches with G. Though in RNA, there's no T, and instead it's replaced with a U. And mRNA is, does virus count as alive? That's a very, it doesn't count as alive yet. That may change in the near future though. As for me, I'm not too sure about it, but we'll see where it goes in the future. Yeah, like a lot of scientists are arguing about it. All right, so mRNA is the actual recipe itself for the proteins, and it passes through the nuclear envelope, which is basically just two phospholipid bilayers, and it goes into the cytoplasm. And now outside of the nucleus, the mRNA finds a ribosome where it can attach it, and it can finally start to manufacture a protein. As for where these ribosomes really are, some of them are just floating around in the cytoplasm, though some of them are attached to this thing called the endoplasmic reticulum, which is a stupid long name, and it basically helps to manufacture proteins. All right, now, enzymes. Enzymes are a type of protein that speed up chemical reactions by acting as a catalyst. Chemical reactions in cells must occur very quickly to sustain life. And while enzymes are part of this reaction, it itself is not consumed in the reaction. So it's not actually used and can be used in other chemical reactions after the first one. And they're not speeding the chemical reactions by just like 5 times or 10 times. It's like hundreds or maybe even thousands. So substrate molecules is what we call them, bind to the enzyme's active site, which you can see here in the diagram I have, is where the substrate attaches, and then the chemical reaction happens. And then the substrate is what the enzyme acts on. And once the chemical reaction is occurred, the product molecules are released, so they go out. Notice how the enzymes not change. It's still the same from beginning to end. And products are what the reaction produces. And note that not all enzymes break a single substrate into 2 products. Not all of them just break down. Some of them can also take 2 substrates into 1 product, so it can combine stuff too. And how enzymes work is they work by lowering the activation energy. And the activation energy is the energy required to kickstart the reaction. So without the enzyme, the activation energy is super high, but when the enzyme binds it and lowers it, and you need less energy, which is provided by ATP and maybe body temperature. And when an enzyme binds to the substrate, activation energy is lowered and lots of more reactions happen, basically. Are there any questions about this part? All right. How do enzymes lower activation energy? So I'm going to go back to it. So it doesn't show it here, but when enzymes attach to the substrate, the thing that binds it, it like ever so slightly sort of bends it a little bit, so it's easier to like break it apart or like fuse them together. So it's like a mechanical change, so it like bends it. That is a very long message in the chat. All right, so photosynthesis. So photosynthesis is the process of converting light, so kinetic energy, into chemical / stored energy carried out by producers. And these include some types of bacteria called cyanobacteria, plants, and algae. And photosynthesis uses carbon dioxide, water, and sunlight to make oxygen and sugar, specifically glucose. And pigments such as chlorophyll found in leaves specialize in absorbing certain wavelengths of light. So they use that light to convert to sugar. And chlorophyll A is the main photosynthetic pigment in plants, and it specializes in this sort of bluish greenish color. But as you can see, literally, not all light is absorbed, and the light that isn't absorbed is reflected. And since plant pigments do not absorb green light, we perceive plants as green. So like they absorb everything else, like purple, blue, and red, and yellow, and stuff. So that's why we see green in plants. And then here in the diagram, I have a bunch of photosynthetic pigments found in plants. So chlorophyll A, chlorophyll B, and carotenoids. And so what this graph is showing is the efficiency of like absorbing color. As you can see here, like most of them really fall into this bluish violet range. And then when it gets to green, it falls off. That's why we see green. Any questions?
Then how come when in autumn and the leaves die, they look orange? Is it because they can't absorb the other colors anymore?
Yes, I
Okay.
think. I'm not too sure. Are there any other questions? All right. So then more enzymes. So many enzymes have partners called cofactors that help catalyze reactions. And cofactors participate in the reaction to increase enzyme activity. So like some enzymes, they need like a cofactor to work first. And then common cofactors include metal ions and vitamins. That's why you need vitamins. And however, not all chemical reactions occur all the time. For example, you do not want to make insulin all the time because that would be very bad. And enzyme inhibition is one way to prevent unneeded reactions from taking place. And it also saves energy and resources. So that's good. So substrate molecules, we know that they typically bind to the active site of the enzyme, but what if another molecule prevents it? So there's two types of inhibition, competitive inhibition and non-competitive inhibition. So non-competitive inhibition involves the inhibitor, so whatever is inhibiting it, changing the active site's shape. So if you can see here on the diagram, they do not bind to the actual active site. Instead, they bind to the enzyme and change the shape of the enzyme so that it no longer accepts the substrate, so can't react to it. And then competitive inhibition, on the other hand, has another molecule just bind to the active site, so the substrate, which is what's supposed to go there, cannot bind. Any questions about this part? Yeah, you can unmute.
How do substrates determine whether or not to inhibit or non-competitive inhibition if they don't have any DNA?
Not sure what you mean by that.
So, like, how do they know when to go in here and when to not go in here?
The inhibitor or the
Yeah.
substrate?
The
So,
substrate.
okay, so the substrate, so non-competitive inhibition, so like the second one here. So the inhibitor changes the shape of the active site. So if you can see here, normal binding, it's sort of like a square shape. And then because it changes the shape, the substrate no longer fits and it can't activate. For competitive inhibition, there's another molecule that binds to the active site. So it just doesn't let the substrate, just blocks it from the active site.
Okay. Thanks.
No problem. So, and then cellular digestion. So vesicles, which is how things get around in the cell, are like small phospholipid bilayers that serves as like taxis for some substances to get around inside the cell. And they can combine from the cell membrane or even other vesicles. And some vesicles have enzymes inside that catalyze reactions that break down substances like sugar, alcohol, or even parts of the cell itself. And these are called lysosomes. However, not all cells have lysosomes. And an example of that is that most plant cells do not have lysosomes. Instead, cellular digestion occurs into these large central vacuoles, which is like this giant sac of like you see in the middle here. So that's the vacuole. And they also help regulate the size and water balance of plant cells. So this is where the cell wall that we mentioned earlier comes in. So if the vacuole gets too big, there's like a ton of water in it, they'll start to push. If there wasn't a cell wall there, it would start to push against the cell membrane. And at one point it will explode, which for the cell, it will not be good. And you do not want cells generally to explode. And for the cell wall, it's made of glucose and bound into like very, very long chains. So if you've taken like a string, a really long string, you've like tied itself around and around and around your fingers, and it's like really strong, and you try to pull it apart, but you can't. I don't know if that's only happened to me. It's like super strong, and that's basically the cell wall, so it helps to keep it from exploding. And that's how it works. And an organelle, there's another organelle to it called the peroxisome that converts hydrogen peroxide, which is H2O2, which is toxic substance, and it's not good, into harmless water. So it just takes away the hydrogen. Yeah, it might explode, but you don't want like a 50% chance for your cells to explode. That is generally not a good idea. You want it to be 0. All right, any questions about this part? All right. Almost all eukaryotic cells have mitochondria, which extract energy from food. So cellular respiration converts sugars or energy into a form the cell can use for work. And to be clear, lipids can sort of bypass this and go into this like the second, like a later stage of cellular respiration. So that's why you store energy as fat. And the space inside the mitochondria is called the matrix and folds in the mitochondrial membrane, you can see here in this diagram, are called cristae. And these are the sites for the chemical reactions of cellular respiration. So eukaryotes that carry out photosynthesis include plants and some protists. And then these cells have chloroplasts, which are the organelles that actually do photosynthesis. They convert sunlight into sugar, which is food for the cell. And then that sugar then goes to the mitochondria. So are there any questions about this part? So now we have like a bunch of sugar and we need to process it into something we can actually use, which for almost all cells is ATP, adenosine triphosphate. So I have a diagram of it here. So ATP is made up of adenine, ribose, which is a type of sugar, and 3 phosphates. So this long chain at the end. And then when one of the phosphates is taken away, we get 3 things. So ADP, which is basically ATP but with 2 phosphates. And then a lone phosphate is floating around, and energy. This energy is then used to do work, which is nice. So carbohydrates, primarily glucose, provide the energy to recombine the used ADP and the phosphate to make ATP again. But then why do we need ATP in the first place? Why not just cut out the middleman and use glucose for energy instead? So here's my version. This is my explanation for it. So imagine a world where you can only use $100 bills. You go to the store to buy bananas, which cost $1 each. Those are some very cheap bananas. You only want 5 bananas. But with your $100 bills, you can only get multiples of 100 bananas. And we're assuming the cashier doesn't have change, which has happened to me before. And this is basically why glucose isn't used for energy. So you can't use $100 bills every time you go to the store, every time you buy a Coke, every time you buy banana. So a single molecule of glucose has like a ton of energy that one chemical reaction might not use all of it. So one single molecule of glucose provides the energy to make 36 to 38 ATP, the amount depends, which can be distributed efficiently for multiple purposes. So are there any questions about this part? So cell structure and the cytoskeleton. So the cytoskeleton is what supports the shape of the cell, which is similar to the scaffolding of building. Like if you've ever seen like a building constructed, you can see there's like X shape, like crisscross, and there's like crisscross. So before they actually put the concrete, so like the rebar in concrete, have you ever heard of that? So the cytoskeleton is basically a network of like protein tracks and tubules found in eukaryotic cells. It has several functions such as structural support, cell division, cell movement, and transport. When are we going to talk about the immune system? This is a cell biology talk, and unfortunately, I do not have the immune system on here. My apologies. And the cytoskeleton is made up of 3 major components. And they're called microfilaments, intermediate filaments, and microtubules. if I can remember correctly, I think microfilaments are about 7 nanometers thick, intermediate filaments about 10, and then microtubules are about 23. And microfilaments are needed for cell contraction and changes in cell shape. And then intermediate filaments form the strong scaffold for mechanical strength. So it's basically what keeps the cell from like imploding on itself. So basically it's the basic structure. And microtubules are the fun ones. They act as like trackways for moving organelles and vesicles. So that how that works, there's like, do you guys know what a cable car is? So it's like it's a car and it's attached to a cable, and cables can go up and down throughout the track, right? So there's like a protein on the microtubule, and with ATP, it changes the shape so that it takes like, how do I put this? It takes like a step forward, and then when energy runs out, it reverts back to its original shape, and then that makes it take to another step forward. So there's 2 steps for like every ATP, or like however number of ATP. And so that's how they move like vesicles. So the vesicles attached to these proteins, and they walk sort of with every ATP, they walk slowly and slowly until they get to their destination. All right, and these are basically what cilia and flagella are made of, which are the like little hairs and tails you see on bacteria, some other cells as well. They're inside the cilia, flagella, and they bend, which is how they move. So like it's how the flagella, for example, like the little tails, they rotate around and around. So that moves it through water, which is basically how most of organisms are made of. We're 60% water, as humans. And they just move through the water by bending the molecules. But with a molecule, I mean a protein called diamine, for your information. All right, so cell-to-cell communication. And then cells in multicellular organisms must constantly communicate with each other in order for tissues to function. And proteins and carbohydrates protruding from animal cells are crucial for cell communication. So earlier we mentioned blood type. This is one of them. And there are others too. And plant cells mainly communicate through plasmodesmata, which are like little holes in the cell walls that connect plant cells. And nutrients and biochemicals pass through to adjacent cells. So animal cells communicate through gap junctions, the exact same thing as plasmodesmata, but in an animal cell, which I have here as a picture. You see it's like little holes connecting them. And it forms a tunnel of proteins such that materials can flow through quickly. And this is good for maintaining homeostasis, as we mentioned earlier. So if like one cell has like way too much sodium in it, some of it will diffuse to the other cell so that the concentration of sodium maintains relatively the same all the time. Right, since animal cells also have tight and adhering junctions, and like tight junctions are like sewing machine stitches, if you've ever seen those before. And it fuses the cell membranes of adjacent cells and forms a leak-proof barrier. This is used for the small intestines, which inside there's like very acidic stuff that digests food and stuff. And you do not want that leaking into your other body parts, or else you will die. And adhering junctions, on the other hand, are like rivets. They connect cells but do not form a leak-proof barrier. And these, you can see there's like little strings attached to them. They use the cytoskeleton for mechanical support. So are there any questions regarding to these? And that's all I have for today. So thanks for coming. And sorry if this presentation is a little short. If you have any questions, you can ask now or in the chat, and I can sort of revert back to the slide. For what grade are you in? I'm a freshman. If you do want to, I can share slides. You might want to provide me with your email because I have no way of contacting you besides that.
Thank you. Bye.
No problem.
Thank you. Bye.
All right.
Thank you. Bye.
I have a
No
question.
problem. You have a question?
I read somewhere that there's a thing called sexual dysmorphism where females like to choose males with for different animal species that are more vibrant in color. But why does it always have to be like that? Why can't it be the other way around?
I have no idea, nor have I ever heard of that before. Though I'll make sure to look at that.
Okay, thank you.